Greetings, Madam Noriza. I'm Len Sebastian here. I'm from class M7 T34.
Lac operon is actually lactose operon. Operon is required for the transport and metabolism of lactose in E. coli and many other enteric bacteria. But for now, I'm going to only focus E. coli, the prokaryote. Several genes clustered as a group under single control mechanism is called an operon. In this case, there are only two factors involved. First, there is no glucose present. Second, there is only lactose as an energy source. Like us, our body undergoes the process homeostasis, which is regulation of the steady internal, physical, chemical conditions in living organisms based on the external environment. Likewise, E. coli are ensuring the endurance of energy from the surrounding, which only has lactose as the energy source. For that, they need to make specific protein. Gene regulation is the most important part in playing and making for the synthesis of protein. Expression of gene is control, is defined as gene regulation. In this process, E. coli is actually regulating its metabolism, either by adjusting the enzyme activity and by changing the amount of enzyme produced. Let's now focus about the founder. In 1961, French biologists André Leboeuf, Francis Jacob and Jacques Monod made a hypothesis. They introduced the aspect of E. coli nutrition. They grew bacteria on culture medium which has no lactose. The bacteria produced has lactose degrading enzyme. But when those bacteria were switched to a medium with lactose, the bacteria has otherwise product. From that, they discovered more about lac operon. They explored the idea of how lac operon switches on and off. They introduced us the Jacob and Monod model. From this picture, we can see the culture medium. Let's now go to the structure. The lac operon consists of lac I, lac Z, lac Y, lac A, promoter and operator. Lac I is in purple color. It is the regulator gene. It encodes for repressor protein. Promoter is actually green color. It's the site for RNA polymerase to start its transcription. The next is operator, which is in yellow color. It's the site to bind repressor protein. Lac Z is used to hydrolyze lactose to glucose and galactose. Lac Y, it codes for membrane protein. Lactose permease allows cells to import lactose. And lac A, to introduce the galactose site with coenzyme A. It attaches particular chemical group to target molecule. Let's not forget that there are always repressor protein, which you can see here, the black one. Repressor protein is used to block attachment of RNA polymerase on promoter. In this case, there are only two conditions present. First, either the presence of lactose. Second, the absence of lactose. Allolactose is a part of molecule of lactose, and allolactose or lactose interferes with the repressor protein's shape. From that, the repressor protein is obstructed. repressor protein cannot bind operator. From that, RNA polymerase binds the promoter. In this thing, gene translation occurs to make specific protein known as enzyme from polypeptide. Lag mRNA is present, but only temporarily, as it is broken down when the concentration of lactose degrades. In this whole process, the operator state or called as switched on. Well, let's now go and check about how it works in the absence of lactose. First, the repressor protein shape is not interfered as there is no allolactose or lactose present in the surrounding and induces the receptor protein. While affecting and inducing the receptor protein, the repressor protein binds the operator and it's actually blocking the part of RNA polymerase. From this, gene translation cannot occur, so no gene transcription. The whole thing is said to be, the operator is called and known as switched off.